Okay, students, welcome to the second of our lectures on proteins. So in this lecture, we're going to be covering two slightly different topics. We're going to be talking about amino acids as we were last time, but this time we're going to be talking about the acid-base properties of amino acids. And then we're going to think about how amino acids are joined together when they are made into a polypeptide chain to make up a protein. So two slightly different topics we're going to be covering today. Okay, so just a quick recap of what we did last time. I hope you remember, but we talked about building blocks. We saw that many biological molecules are made from simple building blocks, subunits, and the building blocks of protein are amino acids. So I hope that you're very familiar with this sort of diagram by now. Here are a number of amino acids joined together into a long chain. This chain has 50 amino acids, some are much, much longer. And this chain is then folded up to make a protein. And further folding to get the final structure of the protein. Okay, and please do remember just how important proteins are. They are essential for playing many different roles in living organisms. Okay, and then last time we looked at different amino acids, and most of the time we talked about the 20 common amino acids. So I hope you remember this diagram of the amino acids with their names, their 3-letter abbreviation, 1-letter abbreviation, and some of their properties sorted out into different groupings. Okay, so that's what we talked about last time. So today I'm going to go on to continue to think about amino acids, but think of them as in their acid-base chemistry. Okay, so I hope you're very familiar with this sort of diagram by now. Here is a diagram of an amino acid. So here is the alpha carbon, here is the variable R group, here is the amino group, the carboxyl group, and a hydrogen atom. And here is exactly the same thing, but written in a slightly different way. So you'll notice here that the amino group is shown as NH2, and the carboxyl group is COOH. Whereas here, the amino group is NH3 with a positive charge, and the carboxyl group is COO with a negative charge. And the difference, of course, is whether or not a hydrogen ion is associated with these groups or not. Here there is an extra hydrogen ion associated. Here the hydrogen ion has been lost. So in an aqueous solution, in a water solution, at a neutral pH, at pH 7, amino acids are ionized. They are in this form. So the amino group gains a proton and has a positive charge, and the carboxyl group loses a proton and has a negative charge. And this form of the amino acid is called a Zwitter ion. It's the Zwitter ionic form. This strange word, Zwitter ion, is a word that comes from German and means hybrid ion, an ion with two different ionic states, if you like, because we've got both a negatively charged group here and a positively charged group. So this is the Zwitter ionic form of an amino acid, and this is the non-ionic form of an amino acid. So zwitterions can act as either an acid or a base. I hope you remember that the definitions of acid and bases. An acid is a proton donor and a base is a proton acceptor. Okay, so in an amino acid in its zwitterionic form, the proton can leave the amino group. It's a proton donor and it can be accepted by the carboxyl group. It's a proton acceptor. So here we see the zwitterion acting as an acid. The proton is being donated from the amino group. So here we go from NH3, positive charge, to NH2 and a free hydrogen ion. Here is the zwitter ion acting as a base. In this case, it's the carboxyl group, but receives a hydrogen ion and goes from being negatively charged to being COOH without a charge. Okay, so taking the amino acid as the whole molecule, it can act as both an acid and a base. And this property is called being amphoteric. Amino acids are amphoteric molecules. They are able to act as ampholites, which means amphoteric electrolytes. Okay, so an electrolyte is a charged molecule. So you'll see here that different parts of a molecule have a different charge. So here we have a positively charged amino group. Here we have a negatively charged carboxyl group. And this property of amino acids is quite important biologically, as we'll go on to see. Okay, so all amino acids have at least two chemical groups which can release protons. So here is an amino group with a net charge of +1. A proton is released. Sorry, first of all, we're looking at the carboxyl group. So here is the carboxyl group with a hydrogen ion attached to it. The hydrogen ion is released. So now we go from a hydrogen ion on the carboxyl group to no hydrogen ion. So we now have a negative charge. So we've gone from a charge of +1 to 0. And now we see the hydrogen ion being released from the amino group. So we go from NH3 with a positive charge to NH2. And now overall, the molecule has a charge of -1. Okay, so +1, 0, -1. So this property of being able to release two hydrogen ions, two protons, remember that a hydrogen ion and a proton, they're interchangeable names for the same thing. So being able to release two protons means that all amino acids are diprotic. However, some amino acids can also release a hydrogen ion from the R group, and these are called triprotic amino acids. Okay, so all amino acids can release two hydrogen ions, some can release 3. So here is a titration curve of an amino acid. This particular amino acid is glycine. So remember the structure of glycine, we looked at it last time. Glycine is the simplest amino acid. Its R group is a a hydrogen atom, just a hydrogen by itself. Okay, glycine is a diprotic amino acid. It can release 2 protons. So here's the alpha carbon. It's shown as CH2 here because it's got both its normal hydrogen atom attached to it plus the hydrogen that is its R group. Okay, so at very low pH, at acidic pH, the glycine molecule is in this form. It has CH2 and the amino group has 3 protons and a positive charge. So this titration curve is taking a solution of glycine at a strongly acidic pH and adding a strong base to it in small drops at a time. Perhaps some of you have carried out this sort of science experiment in chemistry and measured the pH after each addition. And this is how we find out about buffering. So this is the titration curve of glycine, okay? So we're increasing the amount of sodium hydroxide, the strong base being added, and we're measuring the pH after each addition. And we see that at some points in the curve, one small addition changes the pH a lot. At other points in the curve, one small addition or quite a lot of additions doesn't change the pH very much at all. And that is the buffering effect. Okay, so this is the titration curve of glycine. So what can we learn from this? Well, we can see that there are 2 points of inflection. That's 2 places where the shape of the curve changes. So here we've got a curve rising and flattening. And here we've got a curve flattening and rising. Do you see? And the point that it changes from one type of curve to the other is the point of inflection. And the same thing here, a curve flattening and then a curve flattening and rising. So a second point of inflection here. And these 2 midpoints where the curve changes direction are called the pK. So there is a pK for each proton donor. So here, if we go back to this diagram, the carboxyl group loses its hydrogen ion, its proton, at pK1, okay, on average. Remember that this is measuring the average behavior of a lot of different molecules. So at the midpoint, at the neutral pH, we have this form with a 0 charge. And then again, as the pH is increasing to basic to alkaline pHs, we lose the second proton from the amino group. So we go from NH3 to NH2. Okay, so 2 pKs shown by 2 points of inflection of the curve. So if we actually look at the value of this midpoint, pK1 can be actually given a value and it's 2.34, okay? So this curve changes its place at a pH of 2.34. And pK2 is a pH of 9.60. Okay, can you see on the curve there? That's 9.6. Okay, so we can actually give a pH value for the pK of each of the points of inflection on the curve for each of the protons that is being lost. And that is exactly what you see in a table of amino acid properties. So this is the table from Leninger textbook again, that we looked at last time, but before we only looked at the names and the abbreviations. So now I'm explaining some more of the information on this table. This is just the molecular weight of the amino acid. And you can see that glycine is a small amino acid. It has a relative molecular mass of 75. But this is the bit we're looking at now, pK1 and pK2 and pKa values. Okay, so pK values. pK1 is from the carboxyl group and pK2 from the amino group. And for each amino acid, the value that that proton, the midpoint of that point of inflection is given here, and the second one here. So you can see that for each amino acid, there are slightly different values. Glycine and alanine, similar amino acids, similar but not the same amino acids. Proline, a very different amino acid and a different measurement of its pK. So the pK depends on the particular properties of the amino acid. Okay. So one important point, and one of the reasons why it's important to understand about these pKs, is that they are very important for the action of amino acids when they are part of a protein, especially when they are part of an enzyme protein and are taking part in a catalytic reaction. You'll be looking at enzymes in much more detail later in this course. So I'm not going to go into details now, but the behavior of the amino acids as they lose their protons when they are part of an amino acid happens at a much lower pH than it does for the group itself. So this is just showing what happens when the similar group is not part of an amino acid. So this is acetic acid, which has a COOH group in a very similar way attached to a methyl group. So here the proton is lost at about pH 4.5, whereas when it's part of an amino acid, its pK is 2.34. And in a similar way for the amino group, here methylamine, the amine group here loses its proton at a pK value of about 10.6, and here within the amino acid as a whole it's about 9.6. So this shift of value, this change of properties of the chemical group when it's part of an amino acid is very important in allowing protein catalysts to carry out so many of the important reactions in cells. Okay, so I'll pause for questions there. There's quite a lot of information that I've given you so far, but I hope that you'll think about any questions, and please do use the LMS to ask questions. If you are going to ask a question, please ask it as precisely as possible. It's very difficult for me to know exactly how to answer a question if a student writes, "I don't understand amino acids." What don't they understand? But if somebody says, "Please explain why losing a proton is important for an amino acid," I know exactly why they are puzzled, and I can then help them and answer that question. So please do ask very precise questions if you can. Okay, so I mentioned buffers before, and that is the effect we're seeing as these two curves flatten out. And these blue rectangles indicate the area that the two groups of the amino acid are acting as buffers, approximately 1 pH unit on either side of the pK. So this pK is 2.34. So there is a buffering effect going on between about 1.3 and 3.3. Okay, do you see that? And here, the buffering effect, the pK is 9.6, so buffering between about 8.6 and 10.6. So there's just the buffering range emphasized. So when we're talking about amino acids in a biological system, inside a cell, which has about a pH of about pH 7 most of the time, physiological pH, you can see that these buffering regions are quite a long way from that neutral pH. The glycine is not buffering at pH 7, and neither are most of the other amino acids that we looked at in the table. In fact, there are no amino acids that are able to buffer at pH 7 with either their carboxyl or their amino groups. Okay, so pause and think about what that means. So another thing that we can learn from titration curves like this is the isoelectric point. So that is the point in the middle of the curve here, where the midpoint of the steepest part of the curve, and it's called PI, point isoelectric point. And this is the point where the amino acid has no net charge, no overall charge, where the most of the amino acid molecules are in this form with a zero charge. Okay, and this is the isoelectric point. Iso meaning the same, and electric referring to the electrical charge. So it's the point which positives and negatives cancel each other out, and there is no net charge. So for glycine in our curve here, the isoelectric point is pH 5.97. At lower pHs, below 5.97, the amino acids solution, the many molecules that make up the solution, have a net positive charge. More molecules are in a positively charged state than in a negatively charged state. And at higher pHs, above pH 5.97, there is a net negative charge. Okay. So this is important when we're thinking about the behavior of amino acids in proteins, in things like electrophoresis. In a few weeks, you'll be learning about methods that we can use to separate proteins out. And one of those is electrophoresis, using the charge of the protein molecules to separate different proteins. So remember this fact when we come on to talk about electrophoresis. This slide isn't behaving now. That's better. Okay, so back to the table in Leninger again, and looking at the next column. So here are our two pK values, pK1 and pK2, and the third column is PI, the isoelectric point. And again, you can see that the PI falls in between pK1 and pK2, but it's slightly different for each amino acid. So we're starting to see that these values are different for the different amino acids. So this is measuring these characteristics of amino acids is one way of identifying which amino acids are present in a solution. Okay, again, a little pause for questions. Okay, so I mentioned earlier that triprotic amino acids are also able to release a proton from their R group. The R group is also a proton donor. So the example I have for you here is glutamate or glutamic acid. So you can see here that the curve is somehow similar to the one we saw for glycine, but there's an extra part added into it here, which has sort of changed its shape, stopped it being a simple curve. And this is because of the proton being released from the R group. So if we look up here to see what's happening, we can see here is the alpha carbon of the amino acid. Here is the amino group and the carboxyl group. And for glutamic acid, there is another chain of carbons ending in another carboxyl group. Okay, so what happens as the pH is gradually increasing is that first of all, the proton is released from the carboxyl group of the amino acid that attached directly to the alpha carbon. So we go from COOH to COO minus, and from a charge of +1 to 0. And that's the pK that's reflected here. It happens at a midpoint of 2.19. The next proton to be released is the one from the second carboxyl group on the R group. So pKr reflects the release of the proton here, and the charge goes from 0 to -1. And pKr is 4.25. Okay, and that's what's changed the shape of the curve here. And then the third pK, again, it's the proton being released from the amino group and the charge going from -1 to -2. And the pK2 is 9.67, up at quite a basic, quite an alkaline pH. Okay, so you can see that this curve is somehow more complex. The titration curve of the carboxyl group that's part of the R group overlaps with pK1 and pK2 and changes its shape. So here at 4.25. Here is another titration curve for another amino acid, and this one is histidine. So this is the shape of histidine, and again here we see the release of a proton from the R group. Here the R group is a ring shape, okay? And there is a proton here that can be released. So here we see pK1, pKr, and pK2, again reflecting the release of the protons, but in this case, pKr happens at a pH of 6. And remember that we said that amino acids can buffer approximately 1 pH unit on either side of their pKr. Okay, so pH 6 just about allows buffering at pH 7, at neutral pH, at biological pH, if you like. And histidine is the only amino acid that can actually buffer at pH 7. So there's its buffering arranged, just about reaching to pH 7. So in a biological system, amino acids in solution, histidine is the only one that buffers at pH 7. And you might hear in physiology, for instance, about the protein buffering effect of proteins in the blood, for instance, in hemoglobin. And this is due to the histidine amino acids that are part of that hemoglobin. Histidine gives the dissolved proteins their buffering capabilities. Okay, and another pause for questions on that. Okay, so the next thing I want to talk about today is how amino acids are joined together to make peptides. Okay, so you've seen this diagram before. Here is amino acid 1, alpha carbon, R group attached. Here is amino acid 2, alpha carbon, R group attached. And now, because we're looking at a polypeptide chain, the amino acids are joined together by a chemical bond called a peptide bond. So each amino acid building block is joined to the next one by a peptide bond. So here's one, the next, and so on along the long chain of amino acids. So how is a peptide bond formed? Well, before I talk about that, a word about the words I'm using. So two amino acids joined together is a peptide. It's a dipeptide, a peptide made of 2 amino acids. 3 amino acids is a tripeptide, and so on. We use a number of different words. So peptides is the name given to any chain of amino acids. Oligopeptides or short peptides, less than about 20 amino acids, and polypeptides are longer single chains of amino acids, up to hundreds of amino acids. And a whole protein can be made up of several polypeptide chains. Okay, so I'm going to be talking about peptides a lot, peptides and polypeptides. So please do understand what those words mean. Okay, so here is the general structure of an amino acid again. And the peptide bond joins the carboxyl group of one amino acid to the amino group of the next amino acid. Okay. So here are 2 amino acids next to each other, lined up nicely with the carboxyl group here and the amino group of the next one here. So those 2 bonds join together like that. Okay, let's see that again. Okay, and again. So the bond forms between the carbon of the carboxyl group of one amino acid and the nitrogen of the amino group of the next amino acid. So this bond is the peptide bond. Okay. So you'll notice that there is a water molecule that's released when the two molecules join together. So the oxygen and the hydrogen are released from the carboxyl group, and another hydrogen is released from the amino group. So now we've just got the oxygen and one of the hydrogens left. And of course, oxygen and two hydrogens is water. So H2O is removed. So this is called a condensation reaction because water is lost. So peptide bond formation is a condensation reaction because water is removed. Okay, any questions? So here is a peptide, a chain of amino acids arranged in a linear sequence within the peptide. So here are a number of amino acids, and each peptide bond is illustrated in yellow here. Okay, so we've got one bond here, the next, and the next, and the next. Okay, so this is where the peptide bonds formed to form this polypeptide. So just for a bit of practice, can you identify the amino acids? Notice we've got the R groups shown here, okay, in red. Okay, so I hope that you're working it out. So first of all, which one is it? Serine. Next, just one single hydrogen for the R group, easy. Glycine. Now, what's this one? A ring structure. Do you remember which amino acids have ring structures? That one is tyrosine. A methyl group, CH3 as the R group, is alanine. And finally, a branched carbon chain. Which one's that? Leucine. So we have 5 amino acids arranged in a sequence: serine, glycine, tyrosine, alanine, leucine. And this is what's known as an amino acid sequence. There is a linear sequence of amino acids within the peptide. Okay? A linear sequence, a line of amino acids. And we know which direction to read it in because the two ends are different. At this end, there is an amino group. So this is the amino terminal end. And at this end is a carboxyl group. So this is the carboxy terminal end. Okay, so amino acid sequences are always read from the amino end to the carboxyl end. And you'll understand why that is when you study protein synthesis in molecular biology next semester. So the amino and carboxyl terminal ends at the two opposite ends of the chain, and a directional sequence of amino acids within that chain. The amino acids are joined by peptide bonds, and water is released, as we have already seen. So what is left of the amino acids when they're joined into a polypeptide is called the amino acid residues. Residue, what's left after the water has been lost. Okay, water lost in the condensation reaction, the amino acid residues are what's left making up the polypeptide. Okay, so let's have a look now at some of the characteristics of a polypeptide chain. So here is another diagram of a polypeptide chain. Here are the R groups, and here are the peptide bonds. Okay, so each peptide bond is quite a flat structure, a planar structure. The atoms involved in the peptide bond more or less all lie in the same plane. So each peptide bond is illustrated here with one of these grey squares. In a long peptide chain like this, the R groups are actually sticking out on opposite sides of the chain. So here is one alpha carbon atom, and here is an R group extending below the chain. Here is the next alpha carbon, and its R group is extending above the chain. Okay, below, above, below. This is a large, bulky R group. Above, below, above, below. So the R groups are on alternating sides of the polypeptide backbone, and each peptide bond is like a, well, as it's illustrated here, like a rectangle joined at a corner. And this is very important in the way that polypeptide chains are able to fold up in 3 dimensions. Here is another illustration where I think that you can see the alternating R groups quite easily. So R group 1 here upwards, 2 downwards, 3 upwards. And here the polypeptide chain is shown sort of going in and out of the plane of the paper, sort of like a zigzag chain. And the peptide bond here and the other atoms involved in forming the peptide bond, you can see that it's quite flat. This bit's coming towards you, that bit's going behind, but it's quite a flat structure. Here's another diagram again. So again shown as a grey rectangle joined at the corner where the alpha carbon is. And here we can see that although the peptide bond itself here is quite rigid, it doesn't turn around this bond because of the arrangement of atoms in this around this bond don't allow it to turn. The bonds that join these atoms to the alpha carbon can rotate, and these are called the psi bond, that's this one, and the phi bond, this one, represented by Greek letters psi and phi. And the angle that each of these can rotate at is called the psi rotation and the phi rotation. So each alpha carbon allows the bonds on either side of it to turn in relation to each other. And this is what allows a polypeptide chain to fold up. So here is another diagram, again similarly showing the flat planar polypeptide bond and the alpha carbon allowing rotation. Here we're sort of looking down from above, and the psi and phi bonds allowing rotation. Okay. So here is a diagram showing how bonds like this can fold up. And here we've got two R groups that are quite large. And you can see that as the polypeptide chain rotates near this alpha carbon, and as the amino acid residues are brought close to each other, if they were pushed really closely together, the atoms that are involved in the R groups would start being pushed in together. This is a process which would need a lot of energy. So it tends not to usually happen in nature. It's called steric interference. There's a collision between the space occupied by the atoms of each of the R groups. So this sort of process puts limits on the possible ways that a polypeptide chain can fold up. If you have two large R groups next to each other, they can't be pushed into each other. But if you have two small R groups, they can be pushed a lot closer together than they could if they're large. So this is quite an important thing that helps us to understand how proteins fold up and why a protein folds into certain shapes and not into others. It's quite important to help us to understand the three-dimensional structure of proteins, which is what we'll be continuing to think about in the next lectures. Any questions there? Okay, so that's all that I wanted to cover for now. Just to remind you, do go to the textbooks. If you're looking at these hard copies from the library, in this 1982 edition, chapter 6, called Proteins, covers the peptide bond formation. And if you have the soft copies of the more recent ones, you'll be in chapter 3, which is called Amino Acids, Peptides, and Proteins. So thank you for listening, and you can see me again next time.